Hello and welcome to biology class. I am Shipra from ADARSH MANDIR SHANKAR Mount Abu. For class 12, we are learning a new chapter, that is biotechnology and its application. So first, what is biotechnology? Biotechnology is the branch includes therapeutics, diagnostic and GM crop for agriculture. GM is genetically modified crops for agriculture, processed food, bioremediations, waste treatment and energy production. So in some fields we are discussing the use of biotechnology, like in agriculture. In agriculture, use of fertilizers and chemicals like pesticide, weedicides makes the soil sterile day by day, and at last they convert the soil into the barren soils. So GM crop can be solution for it. So what is genetically modified organism? What is GMO? GMO are the plants, animals, fungi and bacteria whose genes have been altered by manipulation. These are known as genetically modified organisms. And these GM plants have been useful in many ways. They may be for made crops more tolerant to abiotic stresses which are caused by the abiotic factors like cold, like drought conditions. Then second, reduce use of chemicals like pesticide. We can use the pest resistant crop, the insect resistant crop. So reduce the uses of chemicals. Then third one is help to reduce post-harvest losses. Then increased efficiency of minerals uses by plants. These modified plants, they are more efficient for the uses of minerals from the soil. Then last one is enhance nutritional value of food. In this GM food, the nutritional value of food will be increased, like rice, golden rice. Golden rice are rich in vitamin A, and they are formed by the use of biotechnology. For example, BT cotton. In this, BT is the Bacillus thuringiensis. Bacillus thuringiensis is the bacteria, and this bacteria introduced into the cotton crop. And this bacteria produce a toxic proteinous substance, and this toxic substance, it kills the insect larva. Okay, what will be the insect? Insect, that is bollworm, budworms, beetles, flies, mosquitoes. So in the larva stage, this toxin, which is released by the Bacillus thuringiensis, it kills the larva and save the crop. So this toxin gene is known as the crab. Now we come to in the medicine field. In the medicine field, genetically engineered insulin is also formed. What is insulin? First we have to know. Insulin is the hormone which is secreted by the pancreas. And what is pancreas? Pancreas is a mixed gland, and it is present in the small intestine between the two arms of duodenum. Between the two arms of duodenum, pancreas is situated, and outer portion of pancreas, it secretes the enzymatic juice for the digestion. And inner portion of the pancreas contains alpha, beta and gamma cells, and beta cell produce the insulin. And what is the use of insulin? Insulin maintains the sugar level in the blood. Okay, so insulin is very useful for the diabetic patients. And before some years, the insulin injection which are used for the diabetes was extracted from the pancreas of cattle and pigs, but illegally. Is a company, American company, it prepared insulin by the use of E. coli. Okay. So what is E. coli? E. coli is the bacteria. So first the process is, first they isolate the gene which is come from the any mammal, from the pancreas. So pancreas have the gene for insulin hormone. So they isolate the gene, and this gene introduced into the E. coli's plasmid. E. coli's DNA, circular DNA, and with the E. coli circular DNA, this gene is inherited into the next generations, and every 20 minutes E. coli can replicate. So the gene is also replicate with the plasmid of E. coli, and the amount of insulin is increased. Okay, then it will be extracted in the gene into the form of injections. So this company, it is formed the insulin, and insulin is first prepared in a pro-insulin form. Then pro-insulin convert into the insulin, mature insulin. So what is the structure of pro-insulin? Pro-insulin contains 3 chains of polypeptides, that is A chain and B chain and C peptide chain. So A and B chain, they are connected with the disulfide bonds. And C peptide chain, it is removed from the A and B chain and it converts into the mature insulin. In the mature insulin, this is the free C peptide chain. It is isolated from it, and these are the A chain and B chain. A peptide chain and B peptide chain, which are connected by the disulfide bonds. So this is the structure of mature insulin. Now we come to the last one, that is gene therapy. What is gene therapy? If a person is born with a hereditary disease like the sickle cell anemia, hemophilia, Down syndrome, Turner syndrome, these are the hereditary disease, and they can be corrective therapy we can use like the gene therapy, like ADA deficiency. ADA deficiency can be cured by the bone marrow transplantation, and it can be treated by the enzyme replacement therapy. This ADA deficiency, it can be treated by the enzyme replacement therapy, and in this therapy, the functional ADA is given to the patient by the injections, and lymphocytes form inside the body of the patient. Lymphocytes are the WBC, and WBC increase the immunity power of the patient. Okay, so lymphocytes form in the blood of patient, and these lymphocytes are grown in the culture outside the body. Okay, then a functional ADA cDNA is introduced into the these lymphocytes. Okay, now these lymphocytes are known as the GM lymphocytes, genetically modified lymphocytes, and they can be introduced into the cells at early embryonic stage. Okay, so this is the gene therapy. Thank you.